we should get started. So welcome very much to both our speakers. We're starting with Mike Lomas at Bigelow, who's going to tell us about how adaptive biology trumps ocean warming. Over to you, Mike, with thanks.
Awesome. Thank you. Thank you, everyone, for showing up. Let's see. There, helps to not start on the there we go. So thank you, everyone. I'm going to take and spend the next 20 minutes or so talking to you about a data analysis from the Sargasso Sea that was recently published. So if you haven't seen the paper, happy to send it around. But hopefully from the title, you can get the take-home message that, at least in the Sargasso Sea in this recent analysis, it seems that biological adaptability can trump at some level ocean warming. So just to orient everyone, the Sargasso Sea is shown where the red star is. We use it as an example of what we think the broader North Atlantic oligotrophic gyre looks like. That's the yellow arrowed circle where the Sargasso Sea is in the northwest corner. But potentially, it's also representative of the other major oligotrophic gyres. So I don't go so far as to arm wave about what it would look like if this were to apply across the ocean, but it may apply across the ocean with more thought and analysis. So just to get started, to orient everyone on my view of the biological pump now and what our models and first principles suggest might be happening. And since everyone's on the phone is an expert in this, I won't take and belabor it too much, but we start with the seasonal pattern where the mixed layer depth is deep sometime early in the year for the Northern Hemisphere, and it shallows through the course of the warmer summer period. That process injects nutrients into the upper ocean. Those fuel phytoplankton growth, which takes carbon dioxide out of the ocean. Those particles sink, get remineralized, and that process goes along. Our models and first principles suggest that as the upper ocean warms, the depth of that mixing will shallow, thus reducing nutrient inputs, thus reducing the ability of phytoplankton to grow, taking up carbon dioxide, therefore less to sink and get remineralized and effectively shut down the biological carbon. And a satellite analysis published about 12 years or so ago now suggested that the ocean regions, the gyre regions here, 4 of them shown, are suggested that they were expanding in size based upon a low chlorophyll criteria. So the area of the ocean with low chlorophyll was expanding in a areal extent in each of these 4 gyre regions. And the North Atlantic is shown here, the third one from the top, and suggesting that indeed it was doing the same thing. So the question then becomes is, what biological mechanisms might there be to counter this progression of events of reduced nutrients, reduced carbon export, reduced primary production? Well, one biological process that's been posited is that you might actually get a change in the preferential recycling of nutrients that may try to improve the nutrient use efficiency of limiting nutrients, primarily nitrogen and phosphorus, but also iron and other trace metals. There could be changes in the phytoplankton community that result in a change in the biological nutrient use efficiency of the phytoplankton themselves. So down here, the graph at the bottom of this figure is data from another paper we published last year, where we can see that, the cyanobacteria, Prochlorococcus and Synechococcus, have much higher cellular C to P ratios, suggesting that they would be better acclimated to grow in an environment where the nutrients from depth are being reduced in their inputs, the nitrogen and the phosphorus. And the third one that's been positive is a change in the grazer community, shifting towards, maybe salps or other sort of non-specific grazers versus copepods. So the goal of today is to take and look at the Bermuda Atlantic time series study data for the last 30 years and try to look at the biogeochemical patterns in the context of these potential biological adaptation mechanisms. So let's start with the biogeochemistry. The top figure here is the temperature record for the last 30 years. The thin black line is the cruise to cruise record. The red dots are the anomalies from the long-term mean for each cruise. And the thick black line running through them is the long-term increase in temperature. So while overall the temperatures increased by about a degree or so over this 30 years, 80% of that has come during the last decade of 2010 to 2020. Associated with that temperature increase, what we seem to be seeing is that the amplitude of the seasonal mixed layer change, so from the maximum in the winter to the minimum in the summer, seems to be getting shallower, less of a seasonal amplitude in the mixing. It's a bit confounded, as you'll see in the later data, by what's happened here in 2010 and 2011. This was an NAO negative winter, which for us in Bermuda will take and actually lead to much deeper mixing. So we have in the middle of this long-term trend, a short-term pulse of a different climate dynamic. And then so the middle graph is that mixed layer record, and then the bottom one is that difference parameter that I was just talking about. And so here we're looking at the impact of that pattern in the physics on changes in nutrients. So the top graph is nitrates. The second graph is our high sensitivity phosphate measurements. Unfortunately, that only started in 2004. That's why the record is shorter. The thin black line are the cruise to cruise values. The diamonds and the red line are the annual values, and the green dash is just the representation of the pattern that it was not significantly changing, positive, increasing or decreasing between 1990 and 2010. As I said, we had the NEO winter in 2010 and 2011, which did lead to a short-term spike in the nitrate inventories. And then they settled back down over the latter part of the 2010 decade to a value that was about 25 to 30% lower than what it was prior to that in the 1990s to 2010. And we see, albeit truncated on the 1990 to 2010 portion of the graph, a very similar pattern in the high sensitivity phosphate measurements. We've also been measuring dissolved organic phosphorus, because again, at the Sargasso Sea, that's the dominant phosphorus pool. And again, we see a very similar pattern, right? You know, non-changing, 2010 slightly higher values, and then decreasing and settling out to a value that is 25% lower. The impact of this nutrient supply on primary production is as we would expect. 1990 to 2010, primary production values show a lot of variability, but are basically stable. We see a short-term response in 2010 to the nutrient input, and then primary production rates basically fall down and actually decrease more like 40 to 50% from the 1990s to 2010 mean value. So that part of ocean warming, nutrient reduction, nutrient inventory reduction, reduction in primary production seems to actually fit with our first principles. The problem is that our carbon export captured in our surface sediment traps does not follow the anticipated pattern. So this top graph here, this is carbon flux. This is data from the 150 meter sediment trap. But we also have data for 200 meters and 300 meters, and they show the exact same pattern. So one could argue that it's actually even a bit more stochastic in the 2010s decade, but for sure it's not decreasing, carbon export anyway. When we look at phosphorus export in our 150 meter traps, that does take and decrease through and into the 2010s decade. So while carbon export is not, phosphorus export is. So just a real quick inter-talk summary. We do see ocean warming. We do see the reduction in nutrients. We do see the reduction in primary production or organic matter production, but we don't see the reduction in the biological carbon pump. So why might that be? So now we're going to dive into those three possible mechanisms that have been proposed for how biology may adapt to this changing environment. So this first data is for the community composition. All of these data are in the panel A and panel B are collected by flow cytometry. So the top panel are our cyanobacteria. Green is Prochlorococcus, red is Synechococcus. These are, again, our inventories over the upper 100 meters. And you can see that while there is their clear seasonality that continues all the way through the entire record, there's no long-term decline or increase. The middle graph is our pico eukaryotes in the light blue and our nano eukaryotes in the dark blue. And you can see that they too have a bit of seasonality, a bit of randomness, but fairly high values between 2002, the beginning of the record, and 2010, 2011. Following that NAO event, then those values start to actually decrease and they lose a lot of that dynamic swing that you saw early on in the record. So this leads to a fundamentally different partitioning amongst carbon amongst the autotrophic organisms, much more heavily skewed now to the cyanobacteria. While we don't directly measure microplankton, so these are the large diatoms, things like that, we can try to estimate it by looking at a carbon-based estimate from carbon to chlorophyll on a cruise-to-cruise basis. That's the dark line, so we assume that to be our total phytoplankton. And the light green line is the sum of our four groups by flow cytometry. And there's no significant or substantial offset between the two, suggesting that one, we aren't missing a large fraction of the phytoplankton in the microplankton, and they are not becoming more or less abundant over the course of time. So the dynamics in the population really are in these four small populations. Also, the fraction of total particle carbon that is living, shown by this new bottom graph in this blue line, is not changing. It's still maintaining its oscillation between 20 and 60% of the total POC. The physiology of those components, however, is changing between the cyanobacteria and the small eukaryotes, and that has an impact on the stoichiometric ratio of the suspended cestonic organic matter. So the top graph here is the carbon to nitrogen ratio in the suspended particles. Middle graph is carbon to phosphorus, and the bottom one is nitrogen to phosphorus. The blue lines in each graph is the canonical Redfield ratio. The thin black lines are the cruise to cruise data, and the red lines are the annual average data. As been shown in the past, the carbon to phosphorus ratio the carbon to nitrogen ratio really doesn't change much and is not overly different from the Redfield ratio. But the carbon to phosphorus in the middle graph and carbon to nitrogen in the bottom graph are both significantly higher than the Redfield ratio in the Sargasso Sea. And they're non-changing from 1990 to 2010 for both ratios. But as you take and go from 2010 to 2020, particularly in the carbon to phosphorus ratio, you can start to see that that's increasing a little bit. And so we've explored that carbon to phosphorus ratio in the ceston using a number of different methods. This is data from that we present in this paper where we use a trait-based model to try to predict on physiology what the C to P ratio would be. And we see a very similar pattern that one, the values are generally higher than Redfield, and that starting in around 2010 or so, we see an increase in the mean C to P ratio. Although that increase is significant, it's only significant because of a large number of data points. It's still a fairly small change in terms of the overall impact. And other studies in the Sargasso Sea, we can see that phytoplankton will, even the same phytoplankton group will change their carbon to phosphorus ratio under nutrient stress. So just for the time being, look at the green circles, which is our Prochlorococcus. When there are lots of nutrients, as shown by the data points that are high in both phosphorus on the X and carbon on the Y, they hang around the Redfield ratio. As nutrients are depleted, they will continue to decrease their carbon and phosphorus quotas per cell, but at some point they can't decrease carbon any further, but they can still change their phosphorus. So that's how they're going from close to Redfield when they're nutrient replete and much different from Redfield when they are nutrient deplete. So we see the change in the community, we see the change in the ceston. So now we're going to take and look at the carbon, the quality of the carbon that's exported. So here we have in the top graph, the carbon to nitrogen ratio in our sediment trap data, carbon to phosphorus in the middle one, and nitrogen to phosphorus in the bottom one. And again, as with the ceston, the blue lines are Redfield, black lines are cruise to cruise, and red lines are the annual values. C to N doesn't change relative to Redfield. But what we can see, and I will point out that the carbon to phosphorus and the nitrogen phosphorus are on log scales only for visualization purposes, because it's really hard to see it because the change is so dramatic. But starting in about 2010, for both carbon to phosphorus and nitrogen to phosphorus, the ratios of carbon and phosphorus and nitrogen to phosphorus in our sediment traps increased dramatically. And so we've estimated that the vast majority of this change in the 2010s decade in the sediment trap stoichiometric ratios is actually due to recycling of the material as it goes from the base of the euphotic zone at 100 meters to this first sediment trap at 150. Only 20% of this change seems to be tied to a change in the source material, that is the phytoplankton themselves. And this is another paper by Tatsuya Tanaka, who was published last year, which just validates that also. And so on the left, in the mauve or pink color, we have our estimate of the Martin coefficient for phosphorus, and in blue for carbon. And so you can see that the Martin curve for phosphorus is generally higher than carbon, and tends to diverge from carbon by staying a bit higher, while carbon Martin coefficients decrease over time. And you can see the summation of that on the right with the spittle plots for, again, in mauve or pink, the phosphorus being higher than the carbon. So it appears that phosphorus is indeed recycled faster than carbon, and that its recycling rate is continuing to stay high while carbon may be decreasing slightly. We also have data that suggests that the role that the trophic structure plays can be important. And so these are some data that we've collected on various batch cruises over the course of time. This is looking at the stoichiometric ratio of carbon to phosphorus in suspended particle material. This is the first box whisker plot. The second box whisker plot is the material that's caught in the sediment trap. Then the next two box whiskers are small phytoplankton. First one is their body tissue. The second one is their fecal pellet material. The next two are large copepods, again, their body material and fecal material. What it shows is that for copepods, they're actually enriched relative to their food, and the fecal pellets are even more enriched in phosphorus. So it's unlikely that copepods would be the source of that phosphorus deplete exported material. However, when we look at salt fecal pellets, their fecal pellets reflect much more closely the material that is actually hitting in the sediment trap. So it's entirely possible that there are changes in the trophic structure between, let's say, copepod grazers and salt grazers. But this is purely based upon trying to line up the stoichiometries of what they pass, quite literally. And we do see that this is data from Debbie Steinberg's group that at least prior to the 2010 decade, SALPs of two different types were increasing. Unfortunately, we don't have data records for these and analyzed yet that go into the 2010s decade. But what we do have is work that a colleague of mine and their graduate student is working on, is looking at the biomass spectral slope to try to use that as a proxy for changes in the size structure of this grazer community. And so the top graph is our daytime data. At bats, we do daytime tows and nighttime tows. The bottom graph is nighttime. The red boxes are where there is a 10-year moving average decrease, significant decrease in the biomass slope. Green is where there is a 10-year moving window increase in the biomass slope. And so you can see that from 1995 to present, where we have data, there is this oscillation between a decreasing slope and an increasing slope back to a decreasing slope, both in the daytime and in the nighttime tow data. This is quite literally hot off the press. We had a committee meeting a couple weeks ago, and so we're still trying to figure out what this really means, but this does support the idea that the grazer community is changing. So just to reiterate, the take-home messages from a biogeochemical point of view, we see the temperature increasing and we see the reduction in nutrient inputs, reduction in primary production. We don't see the reduction in biological carbon pump activity. We do see evidence from a biological point of view that there is a process that's enhancing the preferential recycling of nutrients. We are seeing biological changes in the phytoplankton community, which will then have an impact of the stoichiometry and the nutrient use efficiency. And while we are seeing changes in the grazer community, we're not quite sure how to interpret them just yet. With that, I will thank the many people over the course of my career who have helped contribute to the thoughts that go into this talk, and I will take any questions from either the chat or in person. Thank you.
Thank you, Mike. Fantastic. Yeah, it's a very intriguing picture. Thank you very much. So I noticed we've already got one question in the chat, if I can remind myself where it is. Here we go. Heidi, do you want to unmute your microphone and ask directly, Heidi?
Yeah, sure. Thanks, Mike. That was fascinating. Actually, now I have 3 questions. I'll ask the first one that I put in the chat, and then if other people don't have questions, I'll come back. In your introduction, you were talking about the time series of shallowing mixed layer depth and the predictions that will continue as warming continues. What's been happening with the nitrocline and phosphocline depths? Are those changing, and are they predicted to change? Because obviously that would affect the interpretation of the mixing impact on nutrient injection into the euphotic zone and the whole biological pump.
Yeah, absolutely. So for the bats data record, they are not significantly over the long term shoaling or deepening, right? Okay. They vary, cruise to cruise, season to season, but there's no long term significant trend. As far as what models are predicting, I can't say that I recall for sure, but I think the implication is that they would, that they would imply that they should be shoaling.
Okay.
But they also, I think a key part of that is most euphotic zones in the gyres are really 2-layer systems. And so the models don't really often treat them as 2-layer systems. So they treat them as a whole. And so, we may find that primary production just decreases, but the partitioning between the upper euphotic and the lower euphotic may actually shift to more primary production slightly deeper, which may actually mute some of that shoaling of the nutricline. But now we're purely in the hypothetical range.
Thanks.
Heidi, I don't have any other questions lined up at present, so if you want to go ahead with your other questions.
Okay, my next question was about the phytoplankton carbon. That was a really cool analysis. And then I was just wondering about the assumptions when you're comparing the total to the cytometric components. You mentioned something about carbon to chlorophyll. So you're measuring chlorophyll on the cruises and then assuming carbon to chlorophyll to estimate the total phytoplankton carbon. Is that right? And if so, are you assuming that carbon to chlorophyll is constant seasonally and through time? And how could you untangle that if it's not?
Sure. Yeah. So no, we don't make any assumptions. So what we did for that analysis is now we took the data for each cruise. So we have discrete samples over the euphotic zone of carbon and chlorophyll. So these are not flow cytometry derived chlorophylls. These are discrete HPLC chlorophyll. Plot them against each other as a bivariate plot, and then take the slope of that so that we remove any of the heterotrophic or detrital carbon so that doesn't impact the carbon to chlorophyll ratio. And then we apply that slope to that cruise, and then we just do that for every single cruise. So we're trying to reflect the carbon to chlorophyll in just the phytoplankton component. Although mixotrophy will be a problem, etc.
I see. So the only assumption is that the offset for heterotrophic component is constant throughout the cruise or something like that.
Well, I mean, because we're not, we take that carbon to the slope of that carbon to chlorophyll and apply it back to chlorophyll. So, so we don't make an assumption about it, but we do have a record that, yeah, sometimes that heterotrophic associated carbon is a bigger fraction of the total than it is other cruises, but that doesn't factor into the calculation. We go right from carbon to chlorophyll to get back to carbon.
Okay, thanks.
Thanks, Heidi. And I see that Ken has a question. Ken, do you want to ask directly?
Yeah, fun stuff. I just started thinking early on in your talk, and then a lot at the end when you talk about SELPs, about the deep traps at OFP, and have you looked at those trends of the same years to see if the ratio of flux changes are the same or more accentuated? What does that look like?
Yeah, so I've looked at them very preliminarily. It's I've not had as ready access to the data as I was hoping to, so that's still an ongoing aspect that I'm trying to pursue more.
Be fun.
So, yeah.
Do we have any more questions? We might just have time for one more, if anybody has one. Heidi, is your third question?
Oh, no, looks like there's a new one though.
Let's give Flavia a go, if that's okay. Just quickly. Flavia, do you want to ask the question directly? Flavia, can you hear me?
Yeah. Was wondering, yes, can you hear me? Can you?
Not very well. Could you possibly type your question into the chat? Just that your voice was breaking up quite badly, so I think it'll probably be easier if you could type the question into the chat, and then Mike can hopefully respond.
Well, Flavia's typing. My third question was very simple, I think. Mike, you talked about the biomass spectrum for the grazers. What was the size range of organisms that were considered in that?
Right. So for bats, we do 4 size classes where the minimum is 200 microns, and it goes up to everything that the net toes capture that's greater than 5 millimeters. So we have a 200 to 500, 500 to 1,000, 1,000 to 5, and 5 to 5,000 and greater. So, you know,
Thanks.
We do see some things bigger than 5 millimeters, but obviously big fish avoid the nets.
Okay, thank you very much. I think Flavia might be having difficulties connecting. So what I'd suggest, Mike, is that if she manages to get back in, I think she's there. If you can answer her question in the chat, because I'm aware we need to move on to Grit's talk.
Yep,
so,
absolutely.
Thank you once again, Mike, for an excellent talk. And I say, cool. People should please feel clear to. Sorry, just put this on mute momentarily.
Yep. No, absolutely. Thank you everyone for listening, and please do feel to reach out if you have questions.